Thank you for joining us today for a grand round to Dr. Stewart for the Division of Endocrinology. So for those of you who aren't aware, November is American Diabetes Month, which makes this talk particularly timely. Dr. Stewart is a professor of medicine, endocrinology, diabetes, and bone disease, as well as director of the Diabetes, Obesity, and Metabolism Institute here at the Icahn School of Medicine at Mount Sinai. He received his medical degree from Columbia, after which he served as a fellow in endocrinology and later a faculty member at Yale and chief of division of endocrinology at the West Haven VA Medical Center. Dr. Stewart later served as chief of the division of endocrinology at the University of Pittsburgh, where he and his team were the first to demonstrate that adult human beta cells could be induced to replicate at substantial rates. He returned to Mount Sinai Hospital 4 years ago in 2012. Dr. Stewart has spent several years studying disorders of bone and mineral metabolism, culminating with a complete definition of the humoral hypercalcemia of malignancy syndrome and purification and sequencing of PTHRP. His most recent research focuses on understanding and developing novel means of introducing the replication and regeneration of insulin-producing pancreatic beta cells. Please join me in welcoming Dr. Stewart.
Thank you, Megan. So this is very fun to be here. I've been here now. This is actually my 4th anniversary. I met with Dennis yesterday, and we celebrated our 4th anniversary. I've got to say, it's an amazing place. I'm having a lot of fun. And so I'm going to take the first couple of moments to brag about, actually, let's see, we've got to go back a couple slides here, hold on a second, to brag about what's happening in diabetes, because I want you to know, as Sinai people, how strong diabetes is. So for the first couple minutes, I just want to tell you about this. So 3 years ago, there were 4 years ago, there were 3 PIs in the world of diabetes here. Now there are 17 PIs and additional research faculty shown here and other PIs over here. So we basically have a huge group of investigators, research funding. This is from July. It's actually now up about 8 million, so it's gone from about 1 million a year to about 8 million a year, and the trajectory is upwards. I wanted to say a word about artificial pancreas. So for those of you who don't know what it is, this is old style. This is 2016 insulin pump and glucose sensor. So the idea is you have a pump, it's got a syringe, you can program it to give sub-Q insulin continuously, as well as boluses before meals. You can also wear a sensor that continuously monitors sub-Q glucose and then transmits that information by Bluetooth or radio frequency to a device that you hold in your pocket called a receiver. And so that's old style. And so basically you had a pump, you had a sensor, and you had information coming from the sensor, and all you needed was a brain to tell the pump what to do. So that's been called historically open loop. This is the closed loop system, or artificial pancreas was just approved by the FDA a couple weeks ago and is now going to be launched by at least one company in the spring. So it's the same thing. There's a pump, it's programmable, it delivers insulin in a way that can be regulated by you. It also has a sensor that talks to a receiver you carry in your pocket. This is what the receiver looks like. It looks like an iPhone. And now all of this can be connected by a radio frequency to this thing, a smartphone, basically a diabetes assistant that basically is continually monitoring your glucose and actually telling the pump what to do. And I won't say more about that, except I just wanted you to know that it's coming, and I wanted you to know that Carol Levy has really spearheaded this on her own at Mount Sinai, and we're the only place in New York City, one of only 2 big cities in the United States that has this. And so this is going to become real in the spring. So pretty exciting. So things that have happened here. So we're the only artificial pancreas program in New York City. We have the largest team of type 1 and type 2 diabetes clinical trial PIs in New York City. We've found the first drugs that make human beta cells replicate. I'm going to talk about that today. We've built a new diabetes obesity clinical research center over at St. Luke's. The U.S. News & World Reports now ranks us as number 11. 5 or 10 years ago, we weren't even ranked. We have now a center grant from NIH, the so-called Diabetes Research Center, shared with Albert Einstein. There are only 16 in the U.S. We have more funding from the Juvenile Diabetes Research Foundation than any other med school in New York. And I won't say any more than that. I just want you to know that if you're at Sinai, you should be proud of diabetes, and also that we're here and we're interested in interacting with everybody who wants to interact. So enough of that. public service announcement. So let's talk about beta cells. So first of all, just a little bit about diabetes. I think everybody knows this. 30 million people in the United States have diabetes. 90% of them are type 2 diabetics. There are another 50 to 80 million who have metabolic syndrome or so-called prediabetes. Globally, the U.S. actually is behind the rest of the world. There are more diabetics in China and India each than there are in the U.S., and it's rampant in other parts of the world, particularly Middle East, Africa, Southeast Asia, et cetera, et cetera. 30% of people in the hospital in the United States, at every hospital, are there because they have diabetes. Diabetes and myocardial infarction, diabetes and angioplasty, diabetes and amputation, diabetes and retinal surgery, diabetes and renal transplant, diabetes and dialysis, et cetera, all the complications of diabetes. So it's a huge issue financially for the United States and for every other country. Annual cost in the U.S. for diabetes is $245 billion. It's obviously a big issue for managed care, et cetera. But what I want to bring home today that you may not know, I assume you know most of this, is that basically all kinds of diabetes are a result at the end of the day of inadequate numbers of pancreatic beta cells that make insulin. So I'm going to tell you a few stories. The first story is just about beta cell mass in type 1 diabetes. And so this is a slide that's made from a paper that came out in 2005. There have now been lots of others. But the point is this. You can measure beta cell mass at autopsy in normal people with normal BMIs. And if you do, somewhere around 1.5% of the pancreas is comprised of beta cells. If you look at people who've had type 1 diabetes for 20 years or 50 years and then died, cardiovascular death, car accident death, et cetera, beta cell mass is reduced. That's not new. That's been known for probably 60, 70, 80 years. What's new is that this number is not zero. Everybody thought it's an autoimmune disease. You have immune attack on every beta cell in your body, and after 2 or 3 years of this, all the beta cells are gone and you've got no more beta cells. And so the significance of these studies, one of which came from UCLA, one of which came from Joslin, and many others since then, is that this number is not zero. Even after 50 years of autoimmunity, you still have pancreatic beta cells. And as people have looked at it more, this number is probably closer to something around 30% of normal. So what do they look like? This is insulin staining of the pancreas in somebody who's had type 1 diabetes for 20 years. And you can see there are insulin-positive cells out in the periphery of the pancreas, and they're adjacent to little pancreatic ductules. And some people think that the ductal cells are the progenitors or stem cells for beta cells. This is an islet from a person with type 1 diabetes for 20 years, and you can see there are still insulin-positive cells in what is otherwise a very sick and unhealthy-looking islet with lots of hyalinization, et cetera. And then you can also see insulin-positive cells in the wall of the, in the epithelium of the pancreatic duct, which is also where some people think beta cells come from. So these kinds of data are sort of prima facie evidence that beta cells are still there 20 years later, and they even look like they're regenerating. Obviously, people only get one autopsy per life, so it's hard to do longitudinal studies like this, but there's some evidence that these actually are also functional. So for example, this is an old slide, but it's from a woman named Lois Jovanovic-Peters in California, and her group, she focuses on young women who have type 1 diabetes, and she's particularly interested in managing them through pregnancy. So if you measure C-peptide, a surrogate for insulin, in women who were pregnant or not yet pregnant but planning to become pregnant in their 20s, C-peptide is undetectable. But in the first few weeks of pregnancy, suddenly they're making insulin again. What happened? What happened is they're immunosuppressed, the autoimmunity is dialed down, and the cells that you saw in their pancreas that are still there are able to increase their insulin production. Now that there are more, actually I was thinking of a different slide. So this is a slide that comes from a renal transplant group in Milan, and they said, look, you know, usual business is we decide you're going to get a kidney, we call you in the middle of the night, we have you come in, and then we pump you full of immunosuppressives, and then slap in a kidney tomorrow, and things turn out pretty well. But maybe things would turn out even better if we pre-treated people with immunosuppression, immunoconditioning with rapamycin. So basically what they did is they took people in the dialysis unit with type 1 diabetes, waiting for a kidney, and divided them into those who would get placebo and those who would get rapamycin. And then they followed them over time and measured C-peptide, again, a surrogate for insulin. And what they found, this was not what they were actually looking for, but this is only part of the study, but what it shows is that if you just take healthy people, not healthy, dialysis patients with type 1 diabetes and put them on rapamycin, suddenly you can see they're making insulin again. So this notion of beta cell regeneration seems plausible. And then now we have a new generation of C-peptide assays that are much more sensitive. And this is showing that even if you've had type 1 diabetes for decades, so this is duration of diabetes. So even if you've had diabetes for 60 years, type 1 diabetes for 60 years, autoimmunity for 60 years, you still have detectable C-peptide. So the notion then in type 1 diabetes is beta cells are still there, autoimmunity doesn't kill all of them, and if you could make them regenerate, you wouldn't have to give them insulin anymore. Unfortunately, type 1 diabetes affects in the US only 2.5 million people. That's not an interesting target for pharma, so until recently, pharma said, that's kind of too bad, we feel really badly for these people, but we're not developing drugs for this. And you'll see that's changing. So let's talk a little bit about type 2 diabetes. This is very much a Sinai story. So this is Rosalind Yalow and Saul Burson, who was chief of medicine here. They got the Nobel Prize in 1977 for the discovery of radioimmunoassay. And one of the first things they looked at was insulin. And what they found to their surprise was that insulin was easily measurable and abundant in the plasma of people with type 2 diabetes. That was surprising because in that era there was just diabetes, and they were all juvenile kids who had diabetes and died, and at autopsy they had no beta cells, so everybody thought type 2 diabetes was just sort of the adult version of that. So when they realized that there was plenty of insulin circulating in people with type 2 diabetes or adult onset diabetes in that era, they developed the notion that people with type 2 diabetes have a different disease. Their problem is that they're resistant to insulin. And so that notion is correct, and it's also pervaded diabetes research for the last 60, 70 years. What's changed, though, is, and if you think about this, that can't be the entire explanation. Everybody who's overweight is insulin resistant, but only about a third of them ever get diabetes. So something else has to happen, and the something else is reduction in beta cell mass. So this is one of, again, many slides from many papers showing this kind of thing. So you can look at the pancreas of non-diabetic, lean people at autopsy, and you see that, again, somewhere around 1.5% of their pancreas is comprised of beta cells. If you look at type 2 diabetic lean people, and that happens, their beta cell mass is also reduced compared to demographically matched, gender matched, et cetera, lean people. If you look at obese people who are not diabetic, beta cell mass is much higher than normals. And if you look at type 2 diabetics who are obese, but otherwise matched gender, age, et cetera, BMI, beta cell mass is reduced by about 60% compared to non-diabetic obese controls, and reduced by about 40% as compared to lean non-diabetic controls. So what this says is what in retrospect it was obvious. There's more to type 2 diabetes. You have to have both insulin resistance and inability to compensate. There's an argument over whether this, so why these guys are high. I would say that most people at this feel that people with obesity who don't get diabetes are genetically endowed with a greater supply of beta cells, and I'll say more about that as we go on. And so these people who have type 2 diabetes were born with fewer beta cells, and as they became insulin resistant, they were unable to mount an appropriate insulin secretory response and therefore became diabetic. So what's the evidence for that kind of thing? So now we're in the era of GWAS studies. There have been huge numbers of GWAS studies in every population. This is a summary of what's been found, but the concept is let's take 100,000 people who don't have diabetes, 100,000 people who do have diabetes, and let's just scan the genomes and find out what genes are involved in the pathogenesis of type 2 diabetes. And we're going to find the liver genes, we'll find the adipocyte genes, we'll find the skeletal muscle genes that are associated with insulin resistance, and then we'll understand what the cause of type 2 diabetes is. Because I guess notice I didn't say it. We don't know what the cause of type 2 diabetes really is. What they found, to everybody's surprise, and it's been repeated again and again and again, is that a lot of the genes that are associated, and some of the most strongly associated ones, are beta cell genes. This is the potassium inward rectifier. That's the target of repaglinide. These are transcription factors. This is a zinc transporter that transports zinc into insulin granules so that beta cells can make insulin crystals, et cetera. So, surprise, surprise, unbiased genomic data trying to find the cause of insulin resistance, in fact, found beta cell genes. So in 2016, I think most people would agree that obviously type 2 diabetes is a strongly genetic disease, and in addition to the insulin resistance component discovered by Burson and Yallo, there's also a strong genetic predisposition that's reflected in reductions in beta cell mass. Okay, so now we know that both type 1 and type 2 diabetes are a result of reductions in beta cell mass, entirely in type 1 diabetes and partly in type 2 diabetes, but importantly. So what do you know about beta cell regeneration? Well, this is a slide, so you can publish in PNAS in 2006 that this is what human islets look like. That's pretty amazing, right? So what the message from this slide is that all of the rest of the world, up until recently, has been studying rodent islets. So pretty much everything we think we know about diabetes and beta cell function is derived from rats and mice and the occasional hamster, and monkey, and dog. It turns out, PNAS 2006, the human islets look nothing like rodent islets. Red are beta cells that make insulin, green are alpha cells that make glucagon, blue are delta cells that make somatostatin, and you can see the architecture is completely different. The arrangement of the cells within the islet is completely different. So here beta cells talk to beta cells, here beta cells talk to alpha cells and somatostatin cells, et cetera. So we have thought for a long time that it was possible to make a rodent that beta cells replicate, and this is a list of genes, proteins, growth factors, et cetera, that are associated with beta cell proliferation. So it turns out there are now many, many ways to make rodent beta cells replicate. This is a list of the ones that work in humans. So we've been studying rodent models for decades and found lots of interesting things, none of which apply to human beta cells. So what do we know about human beta cell replication? Well, this is a graph from a guy named Chris Rhodes, but it's also a composite of data or representative of data from lots of other studies. So it's showing beta cell proliferation rates as a function of age. So this is embryonic life here. This is the first year of life. This is childhood up to puberty. This is young adulthood, and then adulthood. And what you can see is the only time that beta cells replicate in human beings is during the first year of life. And the only time they replicate in meaningful rates is at that rate. And then after the first year of life and early toddlerhood, they tail off. And by the time you're a teenager, it's pretty much over. And in adults, there's very, very little beta cell replication. So this is pretty interesting, right? I mean, there aren't many tissues in embryos and neonates that are only replicating at 2%. That's a really low rate. So even at their maximum, beta cells only replicate at a rate of around 2%. The other important point is you made your beta cells, and if you're not a type 1 diabetic, the beta cells you have now are the beta cells you made when you were 1 year old, and you still have them, and you'll have them, they'll take them with you to the grave. So these kinds of data give a sense of what the physiological rate of replication might be, and they also show us a few problems. One is the only tissues we have to study human beta cell replication in are cadaver pancreas donors, islets, which by their very nature refuse to replicate and have never been able to be induced to replicate. Also, if you ever discovered a drug that was good for beta cell replication, you wouldn't be testing it in 1 year olds, you'd be or 6 months olds, you'd be testing it in people in this age group. So it means that this is challenging. So we got into this when I was still in Pittsburgh about 20 years ago, and I won't go through how we got into this, but it's a story in and of its own. But we just said, look, let's think about this. How could it be that one single cell type refuses to replicate when there are so many ways to make other cells replicate? The answer I'm going to tell you is that you can make them replicate if you give them the right signals. So we thought about it like this. You know, if you got an old truck and the thing won't start, and at least what I would do, I don't know if you would do this, is you'd pop the hood. This is for California people in the room. This is a famous place in the Bay Area. And as you pop the hood, you look inside and see if you got a carburetor, see if you have ignition wires, see if you got all the other stuff. So the beta cell cycle, cell biology counterpart would be to open up beta cells and find out what they have for cell cycle regulatory molecules. So this is sort of a summary of all the cell cycle molecules that are in every cell: yeast, flies, worms, people, etc., and are misregulated in cancer. So we decided to just go and catalog all these guys and see what there are. And we found a few interesting things. First of all, every cell cycle that controls the G1S transition is present in human beta cells. Secondly, all the cell cycle molecules in green are molecules that activate cell cycle, and they're there. They're apparently waiting to go. On the other hand, all the red molecules, these are all cell cycle inhibitors, and so beta cells are richly endowed with cell cycle inhibitors, and that's actually different than most other cells. Most cells have one or two of these, one or two of these, one or two of these, and they're still able to replicate. But beta cells are basically natively overexpressing every cell cycle inhibitor known to man. So basically you've got a car with one foot on the gas, and then the handbrake is on, every other brake you can think of is on in these cells. So we said, let's maybe just pour some gas in the carburetor and see if we can get it to go. Let's take some viruses that overexpress cyclin Ds. There are 3 of them, and their partners, cyclin-dependent kinases 4 and 6, and let's throw them at pancreatic beta cells, at islets from cadaver donors that won't replicate and haven't been shown to replicate, see if we can make them go. So that's what this is. So these are human islets. This one is just infected with nothing, no virus. Green is insulin, so every one of these little green things is a beta cell, and each one of the black holes is a nucleus. If they were replicating, they'd have a red nucleus, but as I've told you, beta cells don't replicate. This is a control virus, also showing it doesn't induce replication. And then if you put in an adenovirus expressing cyclin D1, D2, D3, or combinations of CDK4, cyclin-dependent kinase 4, and D-cyclins, or CDK6 and D-cyclins, you don't have to have a lot of imagination to see that there's a lot of replication. And so this is an important slide because it's the first example, first time anybody's ever seen replication in human beta cells at rates in the range that might be interesting therapeutically. You can count all the red nuclei in all the green cells, and under normal circumstances in the controls, there's essentially no beta cell replication, but you can get beta cell replication rates up to 60 times higher by overexpressing cyclins and CDKs. So this was important because it showed that, guess what, you can make beta cells replicate. They're willing to listen if you give them the right signals. That's the good news. The bad news is no one's doing gene therapy for 30 million people with type 2 diabetes, and nobody's doing gene therapy for 3-year-olds with type 1 diabetes. So we began to think about this some more and say, you know, I wonder if we couldn't develop some sort of pill that would do the same kind of thing. And I'll say at this point that these are ideas we had in Pittsburgh, and we began to build this stuff in Pittsburgh, the cells to do this, what I'm about to show you. But one of the reasons I came to Sinai is that you all had a high throughput screening facility, and that enabled us to do what I'm about to show you. So the work that I'm about to show you is done by this really remarkable guy who's now a research associate professor named Peng Wang. He's the gift that keeps on giving. He does amazing things. And this was published in March last year. So the story begins with something everybody in the room has heard of, MYC. MYC is an oncogene. We all know that MYC is a horrible thing. It's overexpressed in lots of different kinds of lung cancers, myelomas, hematologic malignancies, and lots of others. But God or whoever else you believe in, Trump, that's a joke, right? You're supposed to just seeing if you're awake. Didn't give us MYC so that we could get lung cancer or myeloma in our waning years. It has a reason, and it turns out that MYC is a universal requisite member of proliferation in every cell type. So MYC is an oncogene, and without any doubt, high level and sustained overexpression is a really bad thing. But also MYC is an essential universal growth mediator. And normally MYC is barely expressed in pancreatic beta cells, but in rodents and juvenile human islets, if you make them replicate or study them, you can see that MYC levels increase a little bit. They don't go up 100x like they do in tumors. They go up maybe 2 or 3 or 4x. I'll skip this one, this is more. And then we made viruses to overexpress MYC in human islets, and we showed that sure enough, if you give gentle, mild doses of MYC to mouse, rat, and human beta cells, you can make them proliferate in the ways that I showed you before. So now we know that MYC actually is something that's important and naturally required for beta cell replication and can also induce it. So then we said, let's think about MYC some more. So this is, I promise I'm not going to go deep into basic science, but just stick with me for 1 second and you'll get this. So this is the promoter of the MYC gene. This is the coding region that encodes MYC itself. If you look at the promoter, what you can see right away is that every mitogenic signaling pathway known to human beings is present on this promoter. So SMADs are represented, STATs are represented, NF-kappa B is represented, TCF-beta, that's beta-catenin, and SMADs again, more beta-catenin signaling, Wnt signaling, and some others, AP, that's Jun, Phos, cyclic AMP signaling. So what we realized is that every growth in every cell requires MYC, and the MYC promoter has signals on it that recognize every mitogenic pathway known to humanity. And so therefore, maybe what we could do is just take advantage of this MYC promoter, which seems to be able to sense proliferation in every cell, or nascent proliferation in every cell. And instead of hooking it up to MYC, we'd hook it up to the firefly luciferase gene, so that every time a cell that had this construct in it got hit by something that was mitogenic, it would give off a flash of light. Okay, that's if you're not into basic science, that may sound impressive. In fact, it's pretty pedestrian in the world of screening. So basically, what Peng did was to take the the MYC promoter, hook it up to luciferase, and then we basically brought those cells here to do a high throughput screen. So the idea here is now we have cells, it doesn't matter what they are, but we have some cells, they have this MYC promoter driven luciferase reporter gene in it. We basically bought a couple of drug libraries from pharmaceutical companies that had a total of 100,000 compounds. We were able to screen them robotically in 384 well plates using this luciferase readout. Of the 100,000 compounds, 4,500 actually activated luciferase. We took the ones that gave the strongest signals and showed that they really did activate MYC, and we then said that got us down to 86, and then we took those 86 and we put them into rat islets to see if it would make them replicate. When I say we, it's always somebody else. In this case, it's this amazing Peng Wang. So basically, took 86 compounds, put them into rat islets to see if there's replication, and one compound induced replication, and that's harmine. So harmine is shown here. So screening more than 100,000 compounds, one of them actually worked. So that's the beauty. You can spend several lifetimes trying to do this manually. So the beauty of high throughput screening is just that. So this is the effect of harmine on beta cell proliferation in rats. Obviously, we were interested to know what harmine was and if it worked in humans. So this is what harmine is. It comes from a plant in the Middle East called Syrian rue or harmol. But in the Amazon, people have been smoking a plant called ayahuasca for years, so you can make teas out of ayahuasca and out of harmol and drink them or smoke them, and I'll come back to that in a moment. So that's why it's in these libraries. It was known to have medicinal properties. So this is showing rat islets stained for insulin and treated with vehicle. There's no proliferation. But if you throw harmine at them, you get beautiful proliferation in rat islets. You can even see them, the beta cells dividing, as shown here. But more importantly, for the first time, harmine actually makes human beta cells replicate. So in the control, there's no replication, but in harmine-treated human islets, you can see replication, and again, you can see cells dividing, and you can see it whether you use readouts such as BRDU or others such as Cas67 and others as well. So what about the rate of replication? Well, in rodent islets, rat islets and mouse islets, it gets about 8% of beta cells replicating. In humans, it gets around 1.5% of beta cells replicating. That's not too far different than what happens in 1-year-old children. So what does harmine do? Harmine basically is a small molecule that inhibits this enzyme called DYRK1A. So I went to the chemical biology people and said, hey guys, have you ever heard of DYRK1A? Has anybody looked at its structure? And what do we know about harmine binding to it? And then a wonderful guy named Roberto Sanchez sent me this movie. And so this is the 3D structure of DYRK1A. This is a pocket that harmine fits into. That's normally the pocket that ATP fits into. So by going into this pocket, harmine blocks the ability of this enzyme, this kinase, to phosphorylate its substrates. Okay? Again, this is not something you just don't walk down the hall in most places and find somebody who knows exactly what to do. So for me, this is fun. I'm not going to take you through this, but just to say that we spent a lot of time trying to understand how it works, and we now have a good idea as to how it works. And I'll just say that basically there's a family of transcription factors called NFATs. Normally they're stuck in the cytoplasm because they're phosphorylated. In situations where you'd like to have beta cells replicate, calcineurin, which is a phosphatase, takes these phosphate groups off. NFATs are able to go into the nucleus, bind to promoters of genes associated with proliferation, including MYC, which is what we were screening with, and cyclins and CDKs, and also it turns off cell cycle inhibitors. So basically, NFATs, when they go to the nucleus, activate proliferation. The reason beta cells don't replicate, or one reason, is that NFATs can't stay in the nucleus because this enzyme DYRK1A rephosphorylates them and kicks them back out of the nucleus. So basically, you can think of DYRK1A as the brakes on human beta cell replication. So why does harmine work? It works because it blocks that, because it blocks DYRK1A. So basically, it's permissive. It allows beta cells to replicate. So now we have a drug that makes beta cells replicate. We have a mechanism of action. We know its target very well. Now we can go back to this slide. So this is the physiological rate of beta cell replication. This is the rate induced by harmine. I would say that we can pretty routinely get it up to higher rates than that by now. In my world, many people have claimed that they found a molecule that makes beta cells replicate, only to find that nobody else could replicate that. So part of the fun of this story for us is this group at Novartis has now reproduced this. This group at MIT, Broad, and Joslin has reproduced it. These guys at Vanderbilt have reproduced it. This guy at Penn has reproduced it. And this guy at Stanford has reproduced it. So the fun part of the story is that suddenly now in 2016, there is a class of molecules that make beta cells replicate. I mentioned that harmine is in this plant ayahuasca, and so our paper came out in March, in the beginning of March last year. This was in the New York Times about 2 weeks later. So this is in the jungle. This is a jungle prison in the Amazon, according to the New York Times. And so this is where you get sent. If you're like a really bad person, if you murdered your children, your family, and 14 police officers, you go to this prison. And the good news is it's pretty minimum security because there's nowhere to go. But they decided that as a therapy to try and make these people, to heal these people, they would have them drinking ayahuasca tea, which is hallucinogenic, and all of these people found Jesus and apologized and blah, blah, blah. So for us, this was interesting to read 2 weeks after we report harmine, and I get calls from prisons in South America on a regular basis now as a result of this. But I'm showing it to you because of the humor, but also because it makes the point that one of our problems now is that harmine is not specific for beta cells. This NFAT, calmodulin, calcineurin pathway is present in every cell type. So here's sort of where this story is right now. So harmine and several analogs of it, which we're now collectively calling harmalogs, get it, harmine analogs, acting on DYRK1A are able to induce human beta cells to replicate. Homologs may simultaneously, I didn't show you this, but simultaneously enhance differentiation and survival as well. That's turning out to be true. 1.5 proliferation rate is better than a stick in the eye, but it's not what you'd like to have if you had a type 1 diabetes. So it'd be nice to come up with ways to make replication go at higher rates. And then, as I mentioned, harmine does lots of things to other cell types, and so we need to come up with a way to target this to beta cells. That's a big deal now in our world there's never been a way to target anything to beta cells. You may have noticed that we never do imaging to measure beta cell mass in type 1 diabetics or first-degree relatives of type 1 diabetics who might have diabetes. That's because we have no way to image beta cells. And I showed you that slide of human islets all mixed up with alpha cells and beta cells and delta cells. We really haven't got much information on what's going on in isolated pure populations of beta cells. For example, no one's ever asked what glucose does to pure populations of beta cells from humans because we don't have ways to sort human beta cells that are very good. So now suddenly there hasn't been a reason to target drugs to beta cells. Now there's a big reason, and American Diabetes Association, Juvenile Diabetes Research Foundation, Pharma, and NIDDK have all made beta cell targeting sort of the next holy grail. It turns out at Sinai, though, things are again pretty amazing. So here's harmine sitting in this ATP pocket. Here's another inhibitor of DYRK1A, cleverly called INDI, which stands for inhibitor of DYRK. And you can see that even though their structures are pretty different, they fit into this pocket and interact with the same amino acids in exactly the same way. So together with Roberto Sanchez and Mike DeVita, we said, let's see if we can make some versions of this that are both more potent, either more potent or more specific. So now we know what the structural requirements are for blocking DYRK1A. Let's see if we can make better DYRK1A inhibitors, and also ones that we could target the beta cells. So this is harmine sitting in this pocket, and this is a virtual compound that, well, actually now it does exist, but so Bob and Roberto said, well, maybe we stick a phenyl ring over here, and look at all this empty space over here. Maybe we should stick something up over there like that, or what the heck, as long as we're doing that, let's stick a bunch of things in different places. And I got to say, in case you think I know what I'm talking about, I'm an internist too, I'm an endocrinologist, so this is all new to me. But it's just fun. You walk across the hall and people know this stuff. So now Bob and Roberto have synthesized, I think, 250 different versions of harmine and related compounds. Most of them are toxic and kill beta cells, but a few of them actually work really nicely. And I would say at this point, I mentioned that we can get beta cell replication with harmine up to around 2%. Now with modifications, we can get it routinely up to 5 to 10%, which is absolutely enough for type 1 diabetes. And even 1.5% is enough for type 2 diabetes. If your beta cell mass in type 2 diabetes is down to say 50% of normal, if you got every beta cell to replicate once, that would be enough to fix it. So this in our world is exciting. It means that beta cell regeneration for type 2 is actually a lot closer than for type 1, but it's also feasible for type 1. So I'm going to tell you one more story. So I told you that I came to Sinai. Okay, well, I didn't tell you that. I'm from New York. I'm actually from 4 blocks from here. So I came to Sinai because I wanted to come home. Some old guy when I got here said, oh, you mean like a salmon? You came home to die. And I said, well, not really. I came because I wanted to do some high-throughput screening. And the other thing I wanted to do was I wanted to do some genomics. So if you think about it, big picture, beta cells don't replicate. 15 years ago, beta cells don't replicate. You'd be crazy to try. Then you realize that a bunch of mouse models, you can make it happen. Then you realize that with manipulating cyclins and CDKs, you can make it happen. Then you do this high throughput screen and realize that DYRK1A exists and is a drug target that makes beta cells replicate. So how else could you figure out other targets that would make beta cells replicate? And really because, and I mean this seriously for this crowd, since I'm a clinical endocrinologist, it's just obvious. Why don't you look at insulinomas? Insulinomas know how to replicate. They've been replicating. We've all been studying MEN1 syndrome for the boards for the last 60 years. So why don't you ask them how to replicate? So this is a project they're doing with Eric Schatt and people in his group. So the idea is this. Insulinomas are small. They're very rare. They're benign. And they replicate. Ki67 replication rates in beta cells in insulinomas by definition are in the 2 to 3% range. So they're benign, they're small, and they replicate. They make too much insulin and they cause hypoglycemia. What diabetic doesn't want to know that recipe? So we said, let's begin to collect these things. And so the idea is if you have a little insulinoma, you localize it, you go in laparoscopically, pull it out, this is what they look like, sew up the hole that it was in, and this is what they look like histologically. I think you guys all know this. So we said, well, let's collect a bunch of insulinomas and do all the genomics on them and see what comes out of that. So this is a project that's going on right now. It's actually we submitted to Nature Medicine. It's under review right now. We have to do some revisions. But basically, we now have collected 78 insulinomas from all over New York and actually all over the US. And with Eric Schatz's help and his team's help, we've been able to do sequencing on 38 of them, doing exome-seq on 26, RNA-seq on 26. And I won't show you all of this, but basically there are a few interesting things. Actually, there are thousands of interesting things coming out of this, but just to give you an idea. So this is called a Circos plot. Chromosome 1, chromosome 2, chromosome 3, yada, 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 22, XY. So it's a way to display copy number gain, copy number loss across the genome. So the insulinomas have a pretty interesting signature. Each of these bars is 20%, so 20, 40, 60, 80, 100. So 20% of insulinomas have lost chromosome 11. 30% of insulinomas have gained an extra copy of chromosome 7. In the world of cancer biology, these Circos plots are common, and no other tumor has this signature. In fact, also, these guys are different than these guys. The tumors that have this are different than the tumors that have this. We've also done all the sequencing, and I don't have time to go through all of this, but we've also done RNA-seq and compared them to normal beta cells. There are about 3,700 genes that are differentially expressed, 2,000 increased in insulinomas compared to beta cells, and 1,500 decreased in insulinomas compared to beta cells. So, so there's 3,700 new potential drug targets and pathways for beta cell regeneration. So the RNA-seq guy who's in charge of all this is a really wonderful guy named Boyan Losic. And he walked into my office one day in the midst of this project, this is about a year ago, said, "Hey Andy, you know what? I've done all this differential expression stuff, and I've also done it exon by exon. And it turns out this is, I've looked at differential expression of beta cells and insulinomas, and I found that 14,000 genes are differentially expressed in insulinomas versus beta cells, and I can do it by exons, and there are actually 80,000 exons that are differentially expressed." And so here's the number one. You probably can't see it. Here it is here. It's on chromosome 1, there's a gene called DYRK1A. Have you ever heard of that? That's like, Boyan, are you kidding me? I've been working on this for the last 4 years. The p-value is 6.4 times 10 to the minus 126. So out of this insulinoma project, the fun thing is, there's way more, but we do a high-throughput screen 100,000 compounds, find one molecule, it's a DYRK1A inhibitor, and then now do a totally independent, unrelated, unbiased project, screen 80,000 exons and 26 insulinomas, and find the same thing. So the fun part of this, for me, is that now it proves that insulinoma data mining is a way to find new pathways and targets for beta cell replication. So I'm going to stop there. I just want to acknowledge a few people. So Peng, I've acknowledged already. He's amazing. And Juan Carlos, Adolfo Garcia Ocaña is a professor in the Diabetes Institute. I haven't shown you a lot of the animal stuff we've done, but he's been important for that. Aaron is a lab tech who's now a graduate student here who is now doing bioinformatics grad school and is actually first author with another guy who's somewhere here, this guy Juan Wang. These are the two first authors on that insulinoma story, et cetera, et cetera. Also, this is fun because Eric has been amazing. I should acknowledge Carmen and Boyan are here. Roberto and Bob DeVita I mentioned. Dan Felsenfeld actually did the screening. And then really a bunch of surgeons, and particularly Barry Nabnet has been just amazing in these projects. So this has been a very fun sort of story to watch unfold, and I'm going to stop there and I'd be happy to answer your questions.
Questions down the back.
So this is a very interesting story, and it brings a couple other things to mind. Number one, have you tested harmine or any of its analogs, or anything that would cause beta cells to replicate more? Have you found any of these to be carcinogenic in animals? And the second thing it brings to mind is, are these compounds hallucinogenic enough that you could go out on the street, sell them, and fund all of your work.
Okay, so two questions. The first question, are they oncogenic? Well, I mean, here's what I think about that. Every pathway known in regenerative biology involves oncogenes. There's no way to make something, a tissue regenerate in ways that don't involve cyclin, CDK, MYC, et cetera, PI3 kinase, et cetera. So regenerative pathways and oncogenic pathways are the same. In fact, we've basically learned about beta cell replication by reverse engineering oncology genomics, right? So what does that really mean? Well, like for any other drug, if you want something to regenerate, you don't want to have a permanent mutation that's on for the next 7 years. So MYC is on at huge levels continuously in tumors that are MYC dependent. But in physiology, if you throw glucose at a beta cell, it goes up 2 or 3-fold for a couple hours and then goes back down. Right, so the trick is not. The worry is that any regenerative therapy could be oncogenic. The reality is that's why you test all the drugs for safety in the first place, and there will be doses that are oncogenic probably, and doses that aren't. So you have to figure that out. The exciting part about this is there was never anything to test before. Now there are things. In terms of the street value of harmine, I wasn't kidding about getting calls all the time. I get calls. I mean, the harmine is still part of the popular culture, apparently through South America, so I get calls all the time. Actually, I get emails all the time. And actually, since you and I are about the same age, I was surprised to learn that people in our generation were smoking ayahuasca and getting high on it when we were young. I guess we were in med school studying. So I don't, I guess we missed something that everybody else knew about. I guess there probably is street value. I haven't tested those waters.
So, Andy,
Yeah.
you mentioned that the rapamycin actually had an effect which was positive for proliferation. And that's probably a negative regulator of NFAT, and the harmine seems to be kind of the opposite.
Yeah.
So,
So I think that the key point there is that, first of all, rapamycin is like a really bad thing for beta cells. Just take beta cells in culture, treat them with rapamycin, it kills them. And if you take human islets, transplant them in rodents and give them rapamycin, we did that, it basically kills them, et cetera. So rapamycin is bad. And so the answer for beta cells. So the answer is that this calmodulin, calcineurin, NFAT, DYRK1A pathway is ubiquitous, and it does different things, but it has different jobs in different tissues. Its job in beta cells is to prevent them from growing, and so again, more reason that we have to figure out a way that the world has to figure a way to target things specifically to beta cells.
So it nicely shows why cyclosporin and FK cause
Yeah,
post-transplant
yeah,
diabetes.
yeah.
It's the same pattern.
And yeah.
As the youngest member of the audience, I'll tell you a one-moment Saul Burson story.
Please, yeah. I put that in there hoping somebody would say
Yeah,
something.
I'm glad you did. I was with him in his office to my good luck, and when he was packing to go to the All India Institute, he didn't tell me why he was going, but I assumed that that's part of the ritual of Nobel Prize nomination. The other is aging, as you presented it, and as we know, seems to be the key to type 2 diabetes. Where in the data you have do we get some greater insight into how normal aging produces so much type 2 diabetes?
Well, I mean, lots of reasons. Let me emphasize what I said before the beginning. I'm a world's expert in diabetes. I don't know the cause of type 2 diabetes. Nobody does, right? So, but as it relates to beta cells, I think what's becoming completely clear in my world is that progressive DNA methylation occurs throughout the genome, and sites that are required, for example, in beta cells, sites that are required for beta cell replication, like cyclins, those sites are methylated and decorated with histone marks that basically prevent those genes from being accessible. So DNA methylation and histone marking are certainly part of it. And so the next phase is also trying to undo some of that. I mean, so another way to ask that question for us is, okay, wonderful news, you got 2% of beta cells to replicate. What's wrong with the other 98%? Why won't they respond? And my best guess is that that some cells have NFAT binding sites that are open and accessible, but most of them don't, as relates to cell cycle genes. Hey, John.
So my question is, this promoter for the MYC is really interesting. So you can use this also for screening, for example, cancer drug too, that's right?
In theory, you know, and so we had a lot of negative hits as well, which we didn't pursue, and we kept saying, oh, we should screen those for those would be anti-oncogenic sort of compounds.
And you got the harmine, which are to drug. But I assume that maybe the drug mostly affected the promoter area. You know, direct affected the promoter area when you screen drug.
In the screen,
It's really
so,
hard for me to get
yeah, let me just go quickly back to that. So I think we're saying the same thing. So let me just find that screen. So here's the sliding question. So the idea is, that was Jack the alligator. So here's the promoter, and the beauty of this is that, yes, if you want to play around with CTCFs or something like NF-kappa B or STATs at their binding sites, you'd find things there. But if you found something on a GPCR or a tyrosine kinase that went through MAP kinase and 14 other steps and impacted on this, you would find that too, right? So we weren't looking just for things that interact directly with the promoter. We would find anything in any pathway that was mitogenic, or as you point out, anti-mitogenic.
Yeah. I'm not surprised we found only one. I thought
Well, we found, again, I went through it quickly. We actually found, out of 100,000 compounds, we found 4,500 that stimulated luciferase. When we asked which ones really activated MYC in the cells we were screening in, there were 86. Okay? And of those 86, only one was harmine. Yeah.
People are interested in genomics for looking for resilience. I was wondering if there have been studies of markedly obese people who have perfectly normal insulin looking for genetic markers that might predict that protection.
So trying to link GWAS to people who are so taking people who've had insulin clamps or glucose clamps and showing that their diabetes is principally or entirely due to insulin resistance?
No, looking for people who have no evidence of diabetes, have perfectly normal glucose hemostasis, but who are markedly obese.
So so definitely there's a lot of GWAS data on that kind of thing, right? So there are genes that are definitely in the GWAS data set that are associated with diabetes and others that are not associated with diabetes at all, right? But I mean, whether, I guess, and I'm sure you know this, in the world of GWAS, exactly what the heck all these SNPs really mean and what they're doing is another story. So I'm not aware of specific GWAS hits in, for example, obese people without diabetes that reveal why they wouldn't have diabetes.
Thank you very much. Thank you.
Thanks. Yeah.